Thank you very much. It's a pleasure to be here. I'm Stuart Campbell. I'm an associate professor in biomedical engineering here at Yale. And in 2018, I co-founded Propria, which is a company that works on commercializing technology from my lab that enables us to build human-engineered heart tissues for use primarily in pharmaceutical research. When we started the company, we were focusing on the conundrum that you see here in this slide. There were a number of companies at the time that were developing a new class of therapies that were targeted specifically at heart muscle. These activities begin with work in high-throughput biochemical screens, followed by assays in isolated animal cells, cardiomyocytes. The problem that companies faced was then making the jump to animal models, which are much more expensive, and you can't carry too many genetic compounds into that expensive process. So what we aim to do is actually fill the gap in this development funnel by creating these engineered human heart tissues that provide companies with detailed contractility tests in mechanically loaded tissues. And what we were able to do at Propria was to streamline technology from my lab that enables you to generate thin strips of engineered human myocardium from iPS-derived cardiomyocytes, which are then placed into these scaffolds, which we call myopods. These cassettes that then carry a thin human-based tissue are able to be inserted into the MyoLab system. I'm not sure there we go. The MyoLab analyzer, which then gives you high-resolution isometric twitch contractions. So this enables you to test your drug compound in a human context with very high fidelity results before you have to make the jump to animal models and eventually clinical models. So in Brantford, just a couple of towns over from where we are right now, we now have sort of a lab set up with several of these instruments in parallel. We take in compounds from pharmaceutical companies. We generate these engineered heart tissues. You can see we just placed one of these tissues in its cassette inside of the MyoLab analyzer. And then we're able to flow drug compounds onto these tissues and register in real time the response of the muscle to those compounds. There are a few things that set our technology apart from other engineered human heart tissue systems that are out there. One of them is that these constructs are incredibly thin. So the way that they're engineered makes them the thinnest on the market. If you're trying to test a drug or if you're trying to apply a gene therapy, you need ready access in terms of diffusion to the cells that make up that tissue. The other thing we can do is we can actually stretch these tissues as they're contracting, and we can check how much contractile reserve they have. When you're exercising, your heart receives more blood back from the peripheral tissues, which causes the heart to stretch, and then the heart has to contract harder to deal with that increased blood flow. We can actually characterize the sensitivity of the tissue in combination with drug treatments and expose responses to drugs that may not be otherwise obvious. So I'll skip ahead here. We now have the capacity to test multiple drugs at the same time in our facility in Brantford, and have at any given time a lot of parallel ongoing studies with major drug companies. This is an example of some of the data that we can get that exposes differences in drugs. These, you'll see the red data and the blue data are responses to two different drugs, both of which are in clinical trials right now, for increasing the contractile strength of heart muscle. By stretching our tissues while testing the effects of these two drugs, we actually were able to expose the fact that one of these drugs enhances the stretch sensitivity of cardiac muscle, while the other actually does the opposite. And it now appears that the drug shown in red is outpacing the other in terms of clinical efficacy. So if you could find that out earlier in the pipeline, that's obviously very valuable. So as I mentioned, our primary users of the technology right now are pharmaceutical companies that are sending us their drugs to validate their performance and efficacy. We're also beginning to grow rapidly in the arena of cardiac gene therapy development. So we recently had one of our partners present the first data available in terms of testing a cardiac gene therapy for an inherited cardiomyopathy in vitro. And finally, we're starting to grow into the area of cardiotoxicity. So using this system to be able to detect cardiotoxic effects of new compounds. We're not currently seeking investment, but we're always looking for other companies to partner with, pharmaceutical companies or other biotech companies. Be happy to talk. Thank you.